Welcome to all B.Sc. second year students of Fishery Science. During last period, we have completed or seen the first part of fish disease management paper, that is the first part of gas bubble disease, GBD. Yes, last part or the first part, GBD, gas bubble disease. This is the disease which is to be caused when excess concentration of gas, when gas it can be saturated in large quantities, more quantities, then this gas, these are to be created, this gas, these are to be create the problem in fishes, and which is to be harmful, which is to be affect or infected the fishes, which is to be have seen in last period. Now today we have seen the next part of this gas bubble disease, that is the symptoms, preventive controls and their causative reasons why this disease these are to be occur. The remaining part of this gas bubble disease we have completed today. Yes, that is the first part we have seen, or the first point, symptoms of gas bubble disease. Which are the symptoms which is to be occur, which are the symptoms which is to be found on the fish body. The symptoms of gas bubble disease. Affected fish may display acute morbidity. What is the first symptoms? Affected fishes may display acute morbidity. What is by morbidity? That is illness. When fish these are to be infected, when fish these are to be affected due to the gas bubble disease, fish is to be illness. Illness, its behavior is to be ill condition. Normal behavior it is to be not behave. And when it is to be illness is to be occur, and mortality, death. Fishes these are to be die, these are to be dead. Fishes these are to be die, these are to be dead. When illness and mortality these are to be occur in fishes, it is the first symptom due to the gas bubble disease, that is illness and mortality of death these are to be seen, these are to be occur. Fish eggs can tolerate a relatively high total gas percentage. Fish eggs can be tolerate. The fish eggs these are to be tolerate, they can be relatively high total gas pressure. Total gas pressure of 110% to 115%. Those eggs of fishes which is to present, these eggs of fishes, they can tolerate, they can live healthy because they can relatively high total gas pressure. It is to be up to how much? 110 to 115% of the gas pressure, and in such gas pressure these eggs these are to be tolerate, these are to be live. After the egg hatches, however, they are very sensitive to the supersaturation of damage. These eggs these are to be very sensitive to the supersaturation damage with mortalities expected at gas. Which is the gas? That is the nitrogen pressure ranging from 102 to 105. When nitrogen gas these are to be increased to 102 to 105. When this nitrogen gas these are to be increased, these are to be more in concentration, and when these gas these are to be increased in supersaturation, it is to be 102 to 105. Then this supersaturation of nitrogen gas these are to be affect, and eggs these are to be more sensitive, and fishes these are to be, or eggs these are to be, mortality these are to be occur, fishes these are to be destroyed, these are to be destroyed condition. For that purpose this nitrogen gas these are more dangerous for this gas bubble disease. Adult fishes are more tolerant of excessive total gas pressure condition. Adult fishes are more tolerant of excessive total gas pressure condition. Those fishes these are to be occur in water bodies, those fishes these are to be occur in ponds or lake. These fishes are better way. Adult fishes are more tolerant of excessive total gas pressure condition on the order of 115 to 120%. on the order of 15 to 120%. Fishes, these are to be tolerate, these are to be a excessive total gas pressure condition which is to present. But when this total gas condition, these are to be 115 to 120%, then fishes, these are to be live fishes, these are to be found healthy condition. Problems can arise. When problems can arise in fishes, when this nitrogen gas, these are to be saturation from 105 to 140% saturation. When the fishes, when these fishes, juveniles, larvae condition, adult condition, these are to be found, and problems can arise at 15 to 15% saturation and at 140% saturation. At highest fishes, these are to be occur, and then fishes, these are to be killed, then fishes, these are to be die. When this nitrogen saturation, these are to be increased up to 105 to 140%, 105 to 140%, then fishes, these are to be die, then fishes, these are to be killed out. Means this nitrogen saturation, these are to be up to ranging how much? 110 to 105, these are to be suitable, these are to be better. But suppose this natural nitrogen saturation, these are to be increased to 105, then it can be kill the fishes, and heavy mortality, these are to be occur in ponds, lakes, or the culture water bodies. This is the important symptom which is to be seen, which is to be occur. See, this is the fish, that is the fish which is to be shown that gas bubble disease a fish. See, this is the a fish which is to be shown that in this gas bubble condition, in this gas bubble region, here is gas bubble condition, these are to be seen. Here is a abnormal behavior, abnormal swelling, these are to be occur on the body, and this abnormal behavior, these are to be cause the illness of the fish. And when fish, these are to be illness, when fish, these are to be ill condition, then it is to be affect, and when it affect, it can be ultimately killed, ultimately die, and heavy mortality, these are to be occur, these are to be seen in culture conditions of water bodies, culture conditions of the water, culture conditions of the water body. Then this gas bubble disease, how these are to be prevented? This gas bubble disease, how these are to be curing, or these are to be preventing the gas bubble disease we have seen here. Yes, the gas bubbles can form in the body cavities of fish, such as behind the eyes, causing exophthalmia, or between layers of the skin. Where gas bubble, these are to be created, gas bubbles can form in the body cavities of fish, and where it is to be formed, it is to be found in the behind the eyes, causing exophthalmia, or between layers of the skin, exophthalmia of eyes, these are to be bulging. Bubbles can form in the thin tissue between the fins or anal, along the back and tail, along the back and tail. Bubbles, these are to be occur. Bubbles can be seen in tissues between fins, between along the back, along the tail. Small bubbles can form within the vascular system. Small bubbles can within the vascular system, blocking the flow of blood and cause the tissue death. When blood flow, these are to be stopped, blocked, then tissue, these are to be die, ultimately fish is to be death. Worse bubbles can form in the gill lamellae and block blood flow. Worse means maximum worse condition, these are to be occur. Bubbles, these are to be occur on gill lamellae, and gill lamellae, these are to be swells, bubbles, block blood flow, and occasionally resulting in death by asphyxiation. Suppose gill lamellae, these are to be damaged, gill lamellae, these are to be broken, these are to be choked up by bloods, and when blood, these are to be not pass in gills, then ultimately due to the insufficient oxygen, dissolved oxygen, or in low dissolved oxygen level, fishes can asphyxiation, गुदमरणे ज्याला आपण म्हणतो. asphyxiation म्हणजे काय? गुदमरणे. Fishes, these are to be die, these are to be dead due to the asphyxiation. Blisters are clearly visible beneath the skin. Blisters on the body, a bloody boils, a bloody blister like swelling, these are to be appear. These blisters are clearly visible beneath the skin, particularly in the head area, in the eye area, around the eyes area, around the ventral area. Such bulging or blister, these are to be seen, these are to be found in skin, eyes, on different part, different areas. Such blister, these are to be found, such blister, these are to be seen. It is the indication that fish is to be affected, fish is to be infected by the gas bubble disease. Sick fish will make a cracking sound. When fishes these are to be ill, sick in condition, then these fish these are to be create a cracking sound when removed from the water, and the skin will crack if you can run your finger across it. When we have observed, when we have pressed the fish by hands, then its skin is to be cracked down due to the bubbles, because pressure is to be exerted on the skin, and ultimately skin is to be wound, these are to be broken, these are to be tear down, and fish becomes die, fish becomes dead. These are the some symptoms of the that is gas bubble disease. Then diagnosis, how this gas bubble disease these are to be diagnosed. Supersaturation disease can be difficult to confirm since problem visible to the naked eye. The problem these are to be visible to the naked eye. Supersaturation of these disease can be difficult to confirm whether fish is to be a gas bubble disease or not. It is to be visible because problem these are to be seen to the naked eye. Gross lesions are commonly not present when the fish are found. Gross lesions these are to be not seen. Gross swelling these are to be not seen when we have observed the fish observed found. Gill biopsies and wet mount examination of other organs under the microscope may show. Biopsies and wet mount examination. Biopsies, small tissue these are to be taken. Their tissue these are to be then cut down into small slides, and then it is to be like that of the cancer biopsies. Biopsies and wet mount of examination with other organs under microscope, it show the gas bubble or gas emboli within the blood streams. Supersaturation is difficult to the diagnose with water test kit since most kits do not measure dissolved nitrogen. You should consider when those are the available gas kits these are to be found in our medicines, our lab. These gas kits these are to do not maintain the or do not measure the nitrogen, dissolved nitrogen. We have developed that is gas kit of oxygen, gas kit of carbon dioxide, but we do not develop the dissolved nitrogen gas kit which measure a diagnosis of supersaturation of disease for any acute death that cannot be attributed to other water quality disorders or specific pathogens or toxins. Specific pathogens or toxins. Such condition these are to be not seen. Such condition these are to be not found because there is no diagnosis condition. There is no facility to diagnose the dissolved oxygen nitrogen, and those considered to be a diagnosis of supersaturation of disease. Any acute death, 5 fish these are to be die, 5 fish these are to be dead. It cannot be attributed other water fishes these are to be die, fishes these are to be dead due to the other qualities or other abnormal condition, other diseases. It is not reported, it is to be not diagnosed. That's why, or any pathogenic condition, toxic condition, these are to be not seen, these are to be not found, and hence fishes these are to be more danger, more serious when gas bubble disease these are to be occur, because there is no perfect measurement, no perfect device or no perfect counting or diagnostic kit these are to be developed, because it is to be due to the nitrogen, dissolved nitrogen, and when this dissolved nitrogen these are to be not properly measured, how we can measure the that is gas bubble disease? It is to be difficult, but we can guess, we can guessing 5 fish these are to be occur, 5 fish these are to be dead, such condition these are to be seen. With these diagnoses we can take, we can some conclusions for about the treatment. Then which treatment these are to be done about the gas bubble disease? The treatment these are to be essential. Gas bubble disease is rarely reversible because of the damage to the internal organs. Gas bubble disease is rarely reversible because of the damage of the internal organs. When internal organs these are to be damaged, fish become die, and when fish become die, it is reversible effect these are to be seen. Reversible effect these are to be seen. Pressure these are to be increased, fish is to be die. Pressure is to be low, fish is to be in different condition, and it is to be die due to the internal organs these are to be damaged, these are to be a broken down, wounded condition these are to be. Attempt to pop these superficial bubbles do not get to the cause of the problem. That is, attempt to pop these superficial bubbles do not get to cause of the problem and probably do more harm than good, as it causes infection and most likely increase the suffering of the animal. Because these pop of treatment, these are to be essential. Pop of treatment, these are to be done. That is, attempt to pop these superficial bubbles do not get to the cause of the problem and probably do more harm than good, as it causes infection and most likely increase the, that is, increase the surfing of the or suffering of the animal. Those animals, these are to be suffer to the gas bubble. These pop up, these conditions, these are to be seen. These are to be seen. See, this is the gas bubble. These are to be seen. These are to be observed. This is the gas bubble disease. This gas bubble disease, these are to be caused. These are to be seen and it is to be found. It is to be occur on the eye region. Sometimes it is to be found on the skin. Sometimes it is to be found on this snout region. Such condition, these are to be observed. Such condition, these are to be seen in this fish infected area, that is gas. Then what is the prevention measures? How this disease, these are to be prevention. Control measures are the prevention. It is obviously better to avoid a situation than to have to cure one. It is to be avoided, obviously better to avoid a situation than to have to cure ones. Avoid if possible causes of supersaturation. Avoid the nitrogen gas. Avoid the liquid nitrogen or the dissolved nitrogen. Avoid if possible causes of supersaturation. Then avoid, perform an immediate water change to restore the proper gas balance. That is, perform an immediate water change to restore the proper gas balance. Those gases, these are to be produced. Those gases, these are to be occur. These proper gas balance, these are to be done. And this gas balance, these are to be done. These are to be performed. When perform an immediate water change to restore the proper gas balance, or clean away algae and reduce the amount of live plant. Clean away those water bodies, these are to be cultured. Those water bodies, fishes, these are to be cultured. These water bodies, these are to be clean away. Algae, these are to be reduced, and those live plants, those algal blooms, those aquatic weeds, these that is algae, these are to be clean, these are to be live plants. They can be reduce their amount, reduce their quantity, because these live plants and cleaning of this water is the other prevention to control the gas bubble disease. Or reduce lightning to reduce oxygen production by the plants. Reduce the lightning. What is the problem? When lightning, these are to be more, these are to be maximum. When lightning, these are to be more maximum, that is oxygen production, these are to be more, and this oxygen production, these are to be more by plants. Again, these are to be cause the problem. That's why reduce the lightning. When we have cultured the fishes in culture pond, culture site, that is lightning condition, these are to be reduced, ultimately to reduce the oxygen production. And when oxygen production, these are to be reduced, the gas bubble disease is to be controlled, or it is to be reduced. Good aeration is one of the simplest way. Good aeration is one of the simplest way to prevent the gas bubble disease. A good aeration condition, these are to be provided in culture pond, culture sites. Bouncing, splashing, spraying and or escaped culture water through air before it reaches fish will promote the release of supersaturated gas. What is the condition? That is bouncing of condition, bouncing of water, splashing of water, spraying of water and or escaped culture water through air before it reaches fish will promote the release of supersaturation gases. Those gases, these are to be have released, those water which is to be have released into the culture site, into the culture condition, bouncing, splashing, spraying and escaped culture water through air before it reaches to the fish ponds or the culture site will promote the release of supersaturated gases. Supersaturated condition, these are to be done. these are to be seen, and it is to be most interesting, it is to be most done, that is those water in which fishes these are to be cultured, in which fishes these are to be growing in these conditions, spraying, bouncing, flashing, that is then cascading, culture water through before reaches its aeration, these are to be promote a proper condition. Increase the salinity if possible in those water bodies, fishes these are to be cultured in these water bodies, we can increase the salinity if possible, if done, it is possible, you can increase the salinity of water, it is also reduce the gas bubble disease, it is also prevent the gas bubble disease. Again, this is see fish infected due to the gas bubble disease, this is that is the fish which is to be found in hands, palm of hand in a human being, and this fish these are to be show bulging of whole body, and this bulging of whole body it condition that fish is to be ill, not swimming properly freely, and when it is not swimming properly freely and it sit any local or it is to be sit down one place as it is abnormal condition, it indicate that fish is to be infected by the gas bubble disease, and such gas bubble disease fish these are to be seen, these are to be observed, it is the problem which is to be when supersaturation of nitrogen gas these are to be found, these are to be occur, cause the gas bubble disease. And when we have control, when we have done these prevention and control measures to minimize this gas bubble disease, definitely gas bubble disease is also controlled and also cured in culture ponds, in culture tanks. With this we have complete the gas bubble disease occur in fishes, and due to this gas bubble disease we can reduce the fish mortality and we can save our crop. With this we have completed this article. Thank you all of you.